Hi. So, in previous session we have learnt about the complements and we have studied various complement pathway, that is classical pathway, we have studied alternative pathway and lectin pathway. So, let us move on, and with complement I almost completely finished the innate immunity. Now, let us move to the adaptive immunity. So, in adaptive immunity we will study the I will introduce you with antigen, what is antigen and immunogen, okay, and we will also discuss about the B cell, okay, how the B cell produce antibody, okay, what kinds of antibodies are produced, and I am not going to take great detail about the antibody diversity, but I will definitely touch upon how the diversity is achieved in case of B cells, okay, which produce different kinds of antibody. And I will also touch upon the T cell diversity. The T cell for every antigen or for every antigenic determinant there is a specific T cells, okay. So, I will touch upon the diversity component in both B and T cells. I will just explain you the key concepts, although I am not going to take you in more finer molecular detail, because that is not in the scope of this course, okay. And then thereafter we will move on various pathogen, host pathogen interaction. We will take up viruses and then we will take up bacteria, so and so, okay. So, let us begin with adaptive immunity. So, here you know that adaptive immune response is basically depend on the innate immune response, okay. So, you are probably familiar with this slide, probably I have shown you in previous session, but I will repeat you, and here I will emphasize how the adaptive immune response is elicited, okay. You know the cellular component of innate immunity is basically consist of almost all cells of the host, which is non-immune cell, and there are professional immune cells, okay, like macrophages, dendritic cells, and eosinophil, neutrophil, basophils, okay, natural killer cells and all those things. Most of these cells expresses the pattern recognition receptor. You know, now you can understand very well about the pattern recognition receptor. There are several families of pattern recognition receptor, and the overall outcome of activation of this PRR mediated signaling is induction of inflammatory cytokines. So, these pattern recognition receptor basically sense the signature molecule of microbial pathogen, which you know that we call it as a PAMPs, okay. And upon sensing these PAMPs, basically it initiate inflammation. I have discussed several sessions about the inflammation. Now you are very well clear about the inflammation, and then it also activate the innate immune response, means that the microbial pathogen will be basically more actively phagocytosed by the these innate immune cells. So, that I mean to say that activate innate immune response. So, more readily phagocytose, more readily production of hydrolases in the endolysosome or phagosomes, and there will be a more aggressive production of reactive oxygen and nitrogen species. That I mean about this activate innate immune responses, okay. And if it is the virus infection, it will induce the type 1 and type 3 interferon, which basically clear the viruses by various mechanism I have explained you in previous session. how these viruses are cleared by shutting down the metabolic machinery and so on and so forth. Okay. So far everything is good, but if the pathogen is not cleared, then what will happen? So if the innate immune system is not able to clear these kind of infection, then that will trigger the development of more specific immune response, and more specific immune response means there will be a activation of some key lymphocytes. As you can see here, there are two key lymphocytes, that is B cells and T cells. Okay. So these B and T cells will generate a highly specific molecule which will be basically help in clearing or elimination of these microbial pathogen. Okay. And these in case of B cell, these B cells will produce a specific antibody, a highly specific antibody against the microbial pathogen. There will be a repertoire of antibody. It's not only one antibody, there will be a array of antibody which is generated against this microbial pathogen. And once this antibody is generated, so every antibody, there are several kinds of antibody which I will discuss in a subsequent session, the different kinds of antibody. All these antibody has some specific downstream function. So once this antibody is generated, then this microbial pathogen will be eliminated. On another hand, this will be also trigger the development of antigen specific T cells. Okay. So antigen specific T cells, they have also variety of function. In if you look at the T cells, there are two major kinds of T cell. One is Th cells, which I have also discussed in previous session, but I am bringing all those key points which is associated with adaptive immunity. So the T cells are T helper cells and cytotoxic T cells. Okay. And this T helper cells can differentiate into particular T helper cells, for example Th1, Th2, Th17, so on and so forth. Okay. And these particular kind of Th cells will develop a particular kind of immune responses. A very simple, if the Th1 response will be elicited, then there will be a killing of intracellular pathogen. Okay, the killing of intracellular bacteria. Okay. And if there will be a Th2 response against that microbial pathogen, then that will trigger more prominent B cell mediated immune response, antibody production, it may trigger the allergy and so on and so forth. Okay. So this is the more pathogen specific immune response, and these highly pathogen specific immune response, we call it as a adaptive immunity. Okay. So in adaptive immunity, basically we talk about the B cells and T cells. Okay. And how the B cells are developed. As you know that B cells are basically produced in bone marrow, and then they will educate, and then there will be a generation of naive B cells. Okay. And then these B cells will educate, and then in a lymph node, and then it will become a antigen specific immunocompetent B cells. Okay. So let's move on. Here I will also talk about the key players of the immunity, which I have already discussed in when I have taken the cell component. Okay, cell component of immunity. As you know that these are the innate immune cell like neutrophil, eosinophil, basophils and natural killer cells. So they are the key innate immune cells, and adaptive immune cells are B lymphocyte and T lymphocyte. Okay. And here you can see that these cells, the macrophages and the dendritic cells, they are kind of bridge between innate and adaptive immunity. I will tell you how. You know that macrophages and dendritic cells, they can phagocytose the microbial pathogen. Okay. And upon phagocytosis, they can produce the inflammatory cytokines. Okay, so these inflammatory cytokines can recruit other immune cell at the site of infection. On another hand, these macrophages can eat up those microbial pathogen, and then they can present it to the the antigen of these microbe over the cell membrane along with one family of molecule which we call it as a MHC, and macrophages can present the antigen along with MHC class 2 molecule. Okay, and this MHC class 2 molecule along with antigen is needed for the development of T cell mediated immune response. So you can understand these cells are linker between both innate and adaptive immune response. Okay, similarly the dendritic cells. So dendritic cells can also induce innate immune responses in terms of production of innate cytokines. Okay, and they can also process the antigen, process the microbial antigen, and they can present along with MHC class 2 molecule, and they can also induce the differentiation of TH cells. Okay, and depends on type of subtype of TH cells, the immune response will be developed. In addition, you know that all nucleated cells can express MHC class 1 molecule. Okay, all nucleated cells. So here there is a one exception, don't forget, that is RBC. RBC do not present any antigen. Okay, so these all rest of non-immune cells, they express MHC class 1 molecule, and they play a very important role during virus infection. So these MHC class 1 molecule can activate cytotoxic T cells. Okay, and once the cytotoxic T cells will be developed, then these cells will basically clear the viral infection. Okay, or trigger the apoptosis of target cell or death of cells. Okay, so in that way, here you can see that all cells, whether it is an immune cell or non-immune cell, they are basically a linking between innate as well as adaptive immunity. The non-immune cell, how they are how they are involved in innate immunity, you can understand that once this non-immune cells, they are infected with say virus, then these cells can, most of these cells can produce type 1 interferon and to some extent inflammatory cytokine. So in that way they are playing important role in innate immunity. On another hand, these cells can also present the antigen along with MHC class 1 molecule, and then they can activate CD8 T cells or cytotoxic T cells. Okay, so in that way they are also involved in adaptive immunity. So here you can see there is a junction, this is a junctional point between innate and adaptive immunity. Okay, now this is a good time that I should discuss or I should compare both innate and adaptive immunity. Okay, so here you can see that there are some characteristic which is given in the second column, and what kind of innate immunity is there and what kind of adaptive immunity is there. So the first is presence. Okay, the innate immunity is already present in the body. It's not it's always there. You can understand anatomical barrier, chemical barrier. So anything will come, there will be a production of these chemical, mucus, hydrochloric acid, enzymes, reactive oxygen species, reactive nitrogen species. It's always there. Okay, and in case of adaptive immunity, it is developed upon exposure to the foreign substance. What I mean to say that once the pathogen or microbe is entered in the host, then there will be a development of adaptive immune response if it is not cleared by the innate immunity. Okay, specificity, if you see the previously people thought that innate immunity has no specificity, but after learning about the pattern recognition receptor, I can say, or you can understand, that innate immunity do have a broad specificity. They can sense the family of molecule and induce appropriate innate immune response. On another hand, adaptive immunity is highly specific. Okay, I will convince you by this statement in subsequent session, but for time being you can understand that the antibodies which is produced against particular antigen is highly specific. So that's why adaptive immunity has a high specificity. The T cells which is recognizing the antigen along with MHC molecule, they are also highly specific. It's not they don't have a broad specificity. So you can understand that adaptive immunity has a very high specificity. Okay. Response, if you look at the response in case of innate immunity, can fight with any living and non-living foreign entities. It's irrespective of whether it is a living or it is non-living. If the host is exposed to some dust, okay, so that can also elicitate the appropriate innate immune response. For example, more production of mucus, so and so forth. Okay, adaptive immune develop a highly specific immune response if not cleared by the innate immunity. Okay, I have explained you in previous slide that if the innate immune system do not clear the microbial pathogen, then this will trigger the development of adaptive immune response. Response time, in case of innate immunity, the response time is rapid. It is within a minute or maximum to few hours. Okay, but adaptive immunity, development of adaptive immune response takes more time. It depends on what kind of microbial pathogen is there and whether this microbial infection is first time or second time. Okay, so if the host is exposed first time with a particular microbial pathogen, then it will take longer time to develop the adaptive immune response. If the same pathogen is infecting the host second time, then it will take relatively less time. Okay, so but in both case it will take a reasonably good amount of time. It may take a day, a few days to couple of weeks time. Okay, potency, if you see that innate immune response, all those innate immune responses are having a limited potency. For example, blood clotting is also innate immune response. Inducing fever is also innate immune response. Okay, they have a limited effect. Okay, and they have a limited potency. On another hand, adaptive immunity, which is highly specific to the pathogen, they are highly potent. Basically, once the adaptive immune response is appropriate, adaptive immune response is developed, then the microbial infection can be eliminated or decline. Okay, start declining. Time span. So every time the response is generated irrespective of number of exposure. For example, an individual is exposed with say some staphylococcus, okay, Staphylococcus aureus. So the innate immune response against staphylococcus, the first time will be same as the second time. There will be a no change. Okay, but if it is inducing the adaptive immune response, the response time will depend on the type or times of exposure. If it is the first time, then it will take longer time, and if it is a second time, then it will take a shorter time. Okay, so and it also depend on that particular microbial pathogen. In case of adaptive immunity, sometimes this will have a very long remembrance. Okay, or the immune response which is developed first time will stay in the host for longer time, or sometime it will be a shorter time, okay, depends on the microbial pathogen, okay. Another is inheritance. So, innate type of immunity is generally inherited from parent and passed to the offspring, okay. So, basically this is a germline encoded genes are there. Over there, there is a there will be a no change, okay. But in case of adaptive immunity, there is a presence of gene segment which will make a specific type of B cells against particular antigen, okay. So, that may change, but the basic framework is also inherited, but the immune response, the development of immune response may vary to some extent, okay. That what I want to say, that the inheritance in both case there is an inheritance, but the in case of innate immunity, the germline encoded receptor, they do not change, okay. They just whatever germline encoded genes are there, they will express and induce the immune response. But in case of adaptive immunity, there is a gene segments which will rearrange, and then that will develop a particular B cell or T cell response. So, that may change, okay. This is not inherited, okay. The another point is intensity of immune response. So, innate immunity cannot react with equal potency upon repeated exposure to the same pathogen. I have explained you, okay. In case of adaptive immunity, the adaptive immune system can remember the specific pathogen which have encountered before, okay. Again, the innate immunity is present since birth, and adaptive immunity is basically developed during the person's lifetime and can be short lived, okay. It can be short lived, it can be a long lived, okay. So, here I am just trying to explain this point. So, for example, there are mirror image twins, okay. They are having a same genetic makeup, okay, and one individual who lived in very clean environment, and they are not that individual is not exposed to the lot of antigen. On another hand, the another individual that individual is living in a not so clean environment, and that individual is keep on exposing with various microbial pathogen, okay. So, this individual will have a really different set of immunity, okay. It is not although the genetic is same, but the individual who lived in not so clean environment, their immunity is much more educated, okay, much more trained. There is a term known as trained immunity, okay, trained innate immunity also is there. So, that immunity will protect that individual from much more wider range of microbial pathogen, okay. So, that what I want to say that, okay. Allergic reaction, in case of innate immunity, there is a no such kind of term is there. So, we do not say that this is a innate immune response, the allergic reaction, but in case of adaptive immunity, it is basically associated with the allergy is associated with various kind of adaptive immune response, antibody mediated, okay, or T cell mediated. It depends, there are variety of allergic reaction, and all these allergic reactions are basically dependent on adaptive immune response. Memory, innate immunity, they do not have a memory, but these days there is a some term known as trained immunity, okay, or so those school of thoughts say that okay, they do have a some kind of memory, but it is not very clear, okay. In textbook, it is very well documented that the innate immunity, they do not have a memory, okay. But on another hand, adaptive immunity has a very long memory or long term memory that depends on the microbial pathogen, and this memory is very important for for making vaccine or developing immunity against the microbial pathogen. Okay. Another is diversity. So, innate immunity has a limited diversity, but on another hand, adaptive immunity has a huge diversity. There is a endless number of antibody can be produced by the host, okay, or there can be endless number of antigen specific T cells can be generated, okay. So, the adaptive immunity has a huge diversity. Third is complement. So, alternative pathway and lectin pathway is more like an innate immunity, although they have a some level of specificity for the mannose residue in case of lectin pathway. On another hand, the classical pathway is purely, or it has a both component of innate and adaptive immunity. The innate component is this production of these complement protein, and the adaptive component is the activation basically takes place through the antibodies, okay. So, that is the classical pathway. Anatomic and physiological barrier. In case of innate immunity, you know very well there are skin, mucous membrane, there will be an increase in temperature, that is fever, which will inhibit the growth of microbial pathogen, the pH and various chemicals such as hydrochloric acid present in our stomach, and there are some substances produced over the skin, that is certain sebaceous secretion that will change the pH, drastic change the pH, so that the pathogenic microbial pathogen cannot grow, okay. On another hand, the adaptive immune organs are lymph node, spleen or mucus associated lymphoid tissues, okay. Composition of the innate immune system is basically composed of physical, chemical, microbiological. You remember we have discussed about the microbiological barrier, okay, microbiological barrier, okay, and there is a phagocytic cells, okay, the dendritic cells, natural killer cells, and there are various soluble protein. If you remember, there are CRP, C-reactive protein, mannose binding lectin, MBL. So, all these are the basically component of innate immunity. On another hand, the adaptive immunity is basically composed of B lymphocyte and T lymphocyte, okay. Development. So, innate immunity is evolutionary, it is a very old, and it is found in both vertebrate and invertebrates, okay. The adaptive immunity is relatively more new or recent. I have explained you when I have initiated innate immunity, okay. So, adaptive immune system has been developed recently and found only in the vertebrates, okay. Example, there are various example, white blood cells fight with bacteria that cause bacteriolysis, inducing various signatures of inflammation, that is redness, swelling, pain, and there is a increase in temperature, okay. In case of adaptive immunity, the chicken pox vaccination, okay, so that we don't get chicken pox, okay. So, the host is exposed with the chicken pox antigen, so that the host will not develop again chicken pox, okay, because of development of appropriate adaptive immunity, that is B cell and T cell mediated immunity, and that will be remembered by the host, and then they will keep the bacteria of those antibody producing cells, that is B cells and T cells, and in that way the host is protected, okay. So, this is all about the discussion about the innate and adaptive immunity. This is a very good comparison. Now, I have one short video which will basically show you the whole immunity, the innate and adaptive immunity, and then this will be a very helpful in understanding overall this innate and adaptive immunity and the junction between innate and adaptive immunity, you can watch and enjoy.
Every second of your life you are under attack. Billions of bacteria, viruses and fungi are trying to make you their home. So our bodies have developed a super complex little army with guards, soldiers, intelligence, weapons factories and communicators to protect you from, well, dying. For this video, let's assume the immune system has 12 different jobs. For example, kill enemies, communicate, etc. And it has 21 different cells and 2 protein forces. These cells have up to 4 different jobs. Let's assign them. Here are the interactions. Now, let's make this understandable. First of all, let's add colors to the jobs. Now, let's illustrate the cells. The central color represents the main job of the cell, while the surrounding ones represent secondary duties. Now the immune system looks like this. Now the interactions. Isn't this complexity just awesome? For this video, we will only talk about these cells and ignore the rest. So what happens in the case of an infection? It's a beautiful day when suddenly a wild rusty nail appears and you cut yourself. The first barrier of the immune system is breached, your skin. Nearby bacteria seize on the opportunity and enter your wound. They start using up the body's resources and double their numbers about every 20 minutes. At first, they fly under the radar, but when a certain bacteria population is reached, they change their behavior and start to damage the body by changing the environment around them. The immune system has to stop them as fast as possible. First of all, your guard cells, known as macrophages, intervene. They are huge cells that guard every border region of the body. Most of the time, they alone can suffocate an attack because they can devour up to 100 intruders each. They swallow the intruder whole and trap it inside a membrane. Then the enemy gets broken down by enzymes and is killed. On top of that, they cause inflammation by ordering the blood vessels to release water into the battlefield, so fighting becomes easier. You notice this as a very mild swelling. When the macrophages fight for too long, they call in heavy backup by releasing messenger proteins that communicate location and urgency. Neutrophils leave their patrol routes in the blood and move to the battlefield. The neutrophils fight so furiously that they kill healthy cells in the process. On top of that, they generate barriers that trap and kill the bacteria. They are indeed so deadly that they evolved to commit suicide after 5 days to prevent them from causing too much damage. If this is not enough to stop the invasion, the brain of the immune system kicks in. The dendritic cell gets active. It reacts to the signals of the soldiers and starts collecting samples from the enemies. They rip them into pieces and present the parts on their outer layer. Now, the dendritic cell makes a crucial decision. Should they call for antivirus forces that eradicate infected body cells or an army of bacteria killers? In this case, antibacteria forces are necessary. It then travels to the closest lymph node in about a day. Here, billions of helper and killer T cells are waiting to be activated. When T cells are born, they go through a difficult and complicated training process, and only a quarter survives. The surviving cells are equipped with a specific setup. And the dendritic cell is on its way looking for a helper T cell with a setup that's just right. It's looking for a helper T cell that can bind to the parts of the intruders which the dendritic cell has presented on its membrane. When it finally finds one, a chain reaction takes place. The helper T cell is activated. It quickly duplicates thousands of times. Some become memory T cells that stay in the lymph node and will make you practically immune against this enemy. Some travel to the field of battle to help out. And the third group goes on to travel to the center of the lymph node to activate a very powerful weapons factory. Like the T cells, they're born with a specific setup. And when a B cell and a T cell with the same setup meet, hell breaks loose. The B cell duplicates rapidly and starts producing millions of little weapons. They work so hard that they would literally die from exhaustion very fast. Here, helper T cells play another important role. They stimulate the hard-working factories and tell them, "Don't die yet, we still need you. Keep going." This also ensures that the factories die if the infection is over, so the body doesn't waste energy or hurt itself. But what is produced by the B cells? You've heard of them, of course, antibodies. Little proteins that are engineered to bind to the surface of the specific intruder. There are even different kinds of antibodies that have slightly different jobs. The helper T cells tell the plasma cells which type is needed the most in this particular invasion. Millions of them flood the blood and saturate the body. Meanwhile, at the site of infection, the situation is getting dire. The intruders have multiplied in number and start hurting the body. Guard and attack cells fight hard, but also die in the process. Helper T cells support them by ordering them to be more aggressive and to stay alive longer. But without help, they can't overwhelm the bacteria. But now the second line of defense arrives. Billions of antibodies flood the battlefield and disable lots of the intruders, rendering them helpless or killing them in the process. They also stun the bacteria and make them an easy target. Their back is built to connect to killer cells so they can connect and kill the enemy more easily. Macrophages are especially good at gnoming up the bacteria which antibodies have attached to. Now the balance shifts. In a team effort, the infection is wiped out. At this point, millions of body cells have already died. No big deal, the losses are quickly replenished. Most immune cells are now useless, and without the constant signals, they commit suicide, so as not to waste any resources. But some stay behind, the memory cells. If this enemy is encountered ever again in the future, they will be ready for it and probably kill it before you even notice. This was a very simplified explanation of parts of the immune system at work. Can you imagine how complex this system is? Even at this level, when we ignore so many players and all the chemistry. Life is awfully complicated, but if we take the time to understand it, we always encounter endless wonders and great beauty.
So I hope this is a very interesting video. It's a very short and very simple and explaining almost all immunity. You can see that there are involvement of innate immunity, adaptive immunity, and how the T helper cell is stimulating the antigen presenting cells and B cells in order to keep the fight against microbial pathogen. So with this I will stop this session, and in next session I will start with antigens. Okay, we will discuss about the antigen, and in subsequent session I will discuss about the properties of antigen. Thank you very much.